Hi, nice to see all of you here again. Friday, and it is 5 o'clock, and therefore it is time for me to go through another exam paper. Thank you to all of you who have just put very kind comments on my community tab as a result of me explaining why I haven't been doing live streams for the last few weeks. Everything health-wise in the family has all kind of settled down, so here I am back again. I'm going to be off in a few weeks' time for a holiday, but I'm going to carry on as much as I can throughout the summer. So I just wanted to let you know. So here we are going through an AS paper now, and I'm concentrating on AS because I'm kind of guessing that by now practically all A-level students will have had their assessments and their grades will have been decided by their teachers. So I'm focusing on AS. So I am going to show you what I would have written for each answer to each question. And then I'm going to show you the mark schemes and show you very honestly what marks I would have got and what I didn't get, because I didn't get them all, funnily enough. And also, if you are watching this not in live stream land, take the opportunity to pause the video so that you can have a go at the questions, or at least think about what you would have written before I reveal my answer and go through the mark scheme. Okay, so here we are, paper 1, AQA of the 2018 exam series. Let's get going. So we've got the blood circulation system in a mammal here. And we have got to give the letters that represent each of the blood vessels. So aorta, that is the blood vessel that leaves the left ventricle of the heart. So it's leaving the heart. So here am I thinking that it is D. Renal vein, that is the blood vessel that is going to be taking deoxygenated blood away from the kidney, renal, back to the heart, therefore G. And the vena cava is the main vein that brings the blood back to the right the right atrium of the heart, and therefore letter F. So here are the answers. And here's the mark scheme. So I got the 3 marks. Nice, hopefully a nice, relatively easy 3 marks. Name the blood vessels that carry blood to the heart muscle. And that, of course, is the coronary arteries. And you have to get that right. There is only one way to get that mark. So you can see here are the examiners. So on this side, I tend to put the mark scheme. On this side, I tend to put the examiner's comments to help teachers and examiners to mark the paper. This is where you have to do a biological drawing. And they tell you to do it in the box below. Label the following parts of the mitochondria on your drawing. So this is what I drew and how I labelled the mitochondria. Notice that I did a biological drawing. I drew what I saw. I didn't just draw a diagram of a mitochondrion. That would not have got me the marks, or wouldn't have got me many marks, let's put it that way. Oh, some questions already. Dopamine, loving the name. Do I have access to the 2020 paper? I'll be honest, yes, I do, but I don't release the 2020 paper and I don't release the 2019 papers. Teachers in school would rightly gun me down in flames if I released those papers, because they are secure papers that teachers in schools absolutely rely on to use for exams, or some of the questions from them. So I don't give access to those papers. Have I attempted this before? Have I attempted doing live streams before? Oh yes, I've been doing them for a while. I'm not saying I'm professional, but I have been doing them for a while. Anyway, back to this question. So notice that my diagram has taken up at least a good proportion of the box that I've given. I've drawn what I've seen, and I have followed the rules for biological drawings. And they are outlined in this mark scheme. There is no sketching like this. Yeah, you just have continuous lines. There are no hanging lines. In other words, if I have a line joining to this line, it is actually going to join. Sorry, it's actually going to join. It's not just going to have a gap between the two lines. There is no shading and has to look reasonably similar. And I have labelled the crystal and the matrix, and therefore I get 3 out of the 4 marks, because the other 2 things that I should have remembered for a biological drawing, you need to title it and you need to give some idea of the scale. We're given the scale up here, the magnification. So I lost mark 4. Oh, have I attempted this before? Well, yes, Dopamine. In order to be able to produce this this PowerPoint, I have done the questions beforehand. Yeah, I'm not obviously not writing this as I am talking to you. I've pre-prepared it. What I don't do, and I think that's worth you saying, I'm going to be really honest with you. I prepare the PowerPoint. I put up all the mark schemes, but when I come to answer the questions, I write what I would have written, and then I look at the mark scheme to see what I would have got as my marks. All right, so I'm not looking at the mark scheme and just rewording it in a kind of teachery way so that I can go through this with you. I hope that makes sense. It's as good as I can do so that I can go through this really nice and efficiently. Otherwise, you'd spend half your time watching me write, and that is frankly boring. So what is a monomer? I know what a monomer is. It is a small molecule, many of which bonded together make a polymer. I know that that is the definition. Here's what the mark scheme wrote. It is a unit molecule. I didn't write that. I just said it was a small molecule from which larger molecules are made. I actually gave a little bit more information in my answer, but I clearly got the mark. Oh, look, reject. You cannot have just have written, oh, it's an atom, many of which make a polymer, or elements, many of which make a polymer. You have to use the right terminology. Lactulose is a disaccharide. It's 2 monosaccharides, 2 monomers of carbohydrates, of sugars, joined together to form let me start that again. Lactulose is a disaccharide made up of galactose and fructose. Other than both being disaccharides, give one similarity and one difference between lactulose and lactose. You need to know what lactose is made of, and lactose is made of glucose and galactose. So the similarity is that they both contain galactose. The difference is that lactulose contains fructose, whereas lactose contains glucose. And there was the mark scheme, and that was by far the most obvious thing. I'm going to be honest, on this question, I could have possibly also written that they both have glycosidic bonds. But that isn't, there were better things to write. And these 2 things seemed the most obvious and the biggest obvious differences between the 2 molecules, which was why I wrote them. Look, ignore references to hydrolysis and condensation. Yeah, they're both made by condensation reactions via glycosidic bonds. Didn't need to talk about alpha or beta for glucose. You just need to know that it's glucose, and it has to be clear. It has to be clear. I hope that makes sense. So I got those 2 marks. Following digestion and absorption of food, the undigested remains are obviously heading out to the other end of the gut. Let's not put too fine a point on this. And they are processed to form feces in the parts of the intestine below the ileum. In fact, the large intestine is generally where your poo is made. The feces of people with constipation is dry and hard. Constipation can be treated by drinking lactulose. Here is the key statement. Lactulose is soluble, but it is not digested or absorbed in the human intestine. In other words, the undigested remains that get through the ileum, where most of the digestion happens, that lactulose joins that undigested remains. It is there too, because it is not digested, but it is soluble. Use your knowledge of water potential. There is a huge clue in the question as to what they're wanting in the answer, to suggest why lactulose can be used to help people suffering from constipation. Well, fundamentally, if lactulose is there in the gut where all the rest of the undigested remains are, then lactulose, because it is soluble, it is going to lower the water potential of the contents of the gut. That means that the cells lining the gut are far more likely to have a higher water potential. And therefore, water is going to move by osmosis from the higher water potential to the lower water potential, down the water potential gradient. And therefore, those undigested remains are going to be far wetter and far softer. Therefore, the person will no longer be constipated. So there we go. Lowers the water potential. This is an abbreviation that I know you can use in exams, which is why I use it. Therefore, water will move by osmosis into the gut, down the water potential gradient, making the feces more wet and soft. So I got this mark point. The other thing that it's worth me saying is that I clearly show, and I've always done this when I mark exam papers, I clearly show exactly where each mark point has been gained. So here, I think, after this word, I've got this mark point, and right at the end of my explanation, because you have to say that it softens the feces, i.e. no more constipation. So I got the 2 marks. And this question is a maths question. And it's talking about, it's getting your head around orders of magnitude using standard form. Lactulose can also be used to treat people who have too high a concentration of hydrogen ions in their blood. The normal range is from 3.55 times 10 to the negative 8 to 4.47 times 10 to the negative 8. Hopefully, clearly, this is the upper limit of the normal range, 4.47. 10 to the negative 8. A patient was found to have a blood hydrogen ion concentration of 2.82 times 10 to the negative 7. That's a whole order of magnitude higher because it's 10 to the negative 7, not 10 to the negative 8. Think about it. This number, if you wrote it out in full, would have 7 zeros after the decimal point because it's to the negative 8. This one would have 6 zeros. So calculate the minimum percentage decrease required to bring the patient's blood hydrogen ion concentration into the normal range. Here's my little diagram of percentage increase. We are going from a higher number down to a smaller number, and we need to work out the difference, the decrease, and we need to make it a proportion of the original higher number. It's like saying that if something has gone down in price from £1 to 50 pence, there has been a 50% decrease because it is 50 out of the original 100 pennies, a pound. Hope that kind of makes sense. So the minimum percentage decrease is for it to get to the higher level of the normal range. So we have got to do 2.82 times 10 to the negative 7 minus 4.47 times 10 to the negative 8 divided by that decrease as a fraction of, therefore divided by the original larger number, 2.82 times 10 to the negative 7. And you times that by 100. And that, if you do the calculation, gives you 84.15%. The number on the calculator was actually 84.148. So I rounded it up to 84.15. Notice that if you had given 84.1, you got all 3 marks because the number on the calculator was 84.148. If you had rounded it up to 84.2, it would have been incorrect rounding and you would have only got a mark for your working. Worth noting that. This rounding is correct and actually this was allowed. All right, and they also actually allowed just 84. So they allowed, you know, some flexibility into the precision of your answer. So I got the 2 marks. Label and draw and label a single DNA molecule. Ooh, dopamine. I have my first cycle 3 test next week. Is it possible to do a summary video on topic 4? I don't know. Topic 4. So I'm not, dopamine, I'm assuming you're doing AQA. I don't know what you mean by chapter 4. What you might be useful, I've done loads of videos on this YouTube channel, a lot of which are public. So do have a look. I'm not sure what you mean by topic 4. Is that chapter 4 of this, what I would call the standard textbook? That's the one with the curly haired boy looking at a test tube on the front of it. I don't know. But have a look at my YouTube channel. And if not, have a look at my website, which I'll talk about in a minute, www.biologycarol.co.uk, because I will have covered topic 4 on my website, but that does require payment. So anyway, I'll talk about that in a minute. Draw and label single DNA nucleotide. Here it is. We're told that this is a DNA molecule, and that is why my pentose sugar here is accurately named. It's not just pentose sugar, it is deoxyribose, because I know this is a DNA molecule. This is my nitrogenous base, and notice that the phosphate group comes off this point on the deoxyribose, not up here. That may be me being pedantic. Here is the mark scheme, and correct shapes and bonds in the correct position. Can I just say that me putting this at an angle is absolutely makes no difference. The fact that they've drawn it straight, and yeah, the same here. So that doesn't matter. What you have to do is get them off the right points on your pentose sugar, your deoxyribose. Oh my goodness, massive load of notes. You can use various abbreviations for phosphate, but you cannot use the word phosphorus because phosphorus is the element, not the chemical group, PO4 3-. Don't accept pentose, ignore references to sugar, yep. Accept a named base. So I could have put either adenine, guanine, cytosine, or thymine. Ignore labeled bonds, that isn't what was needed. Accept correct shapes with incorrect labels. Okay, all right. Yeah, that's fine. Accept any pentagon. Yes, fair enough. Give 2 features of DNA and explain how each one is important in the semi-conservative replication of DNA. You have got to focus your mind into what are the features of the DNA molecule that specifically help in DNA replication. And there are 3 obvious ones, one of which is the complementary base pairing of the nitrogenous bases, because that causes accurate replication. Does it make sense that once those 2 strands have been separated, that because if we know the order, the base order of one strand, we absolutely know the base order of the second strand of the DNA. So does it make sense that the complementary base pairing brings about accurate, oh, sorry, I didn't put that in, accurate replication? Really useful for you to be able to nail that phrase. Complementary base pairing allows accurate replication. And the other thing is, the other one that I thought of, the next one that I thought of is because DNA is double-stranded, both of those strands can act as a template. The other thing of DNA is the fact that those two strands are held together by hydrogen bonds between those nitrogenous bases, those complementary base pairs. And that's really useful because they are easy to break by DNA helicase. So all of those are in the mark scheme, rightly, and I got the 2 marks for saying those two. Notice that other features of DNA don't aid replication. The fact that DNA is really long, it holds huge amounts of genetic material, genetic information, but that doesn't help DNA replication. The fact that it's got a sugar phosphate backbone that protects those inner bases doesn't help DNA replication. So it's about being able to identify the features that absolutely aid the replication. Okay, so will this, Dr. Cheddar Cheese, loving that name. You've spelled cheddar wrong, but that might be me being pedantic. Anyway, will this be useful for OCR as well? Yes, it will, because it covers the same content. And the A-level questions are similar in their difficulty, as long as I'm assuming that you're doing the paper 2 AS exam. So yes. So I hope that's helpful. So this is talking about the replication of mitochondrial DNA. And here they tell you that with mitochondrial DNA, and I've just drawn a really simple DNA strand, it's a bit weird because the second replication of the second strand of DNA only happens once the first strand has 2/3 of it have been replicated. So I'm kind of guessing that the DNA strands get split. I didn't show that in the diagram. The DNA strands get split. The first one, the enzymes have to replication, DNA polymerase gets 2/3 of the way down the first strand before then the second strand is replicated. So does it make sense that if the whole piece of DNA is 16,500 base pairs long, how long would it take to copy? Does it make sense that we've got to do the 2/3 bit first and then the whole 16,500? So actually, the time that it will take is the time that it will take to replicate, as it were, 27,500 bases or nucleotides. And if that's done at 50 per second, it's 550 seconds. So this is about understanding the implication of this part of the question. And therefore, this is the answer, which gets me the mark. Oh, don't worry. I'm loving the name, So prokaryotic cell. Sorry about me being pedantic. That's being a bit teachery for you, isn't it? So name the structures W to Z. So from the inside out, this is a prokaryotic cell. And I always say to students, if you work from the inside to the outside of the diagram, especially for prokaryotes, you're always going to label each layer correctly. Because from the inside is always going to be the cell surface membrane. Can't remember, I did them all at the same time. Cell surface membrane. The next thing out from that is the cell wall. And the next layer out from that is the slime layer, slime capsule or slime layer. And then we get these, you know, these funny little lines, which are hopefully, obviously, the flagella, and one is the flagellum. And there, you know, this is all AO1. This is knowledge. Do you know the stuff? So I get the 2 marks because I got all 4 correct. This is really common, that the term capsid from viral structure is confused with capsule from bacterial structure or prokaryotic structure. Yeah. Name the main biological molecule in W. So you had to know what W was. Cell surface membrane, the main biological molecule, phospholipid. Name the main biological molecule in the cell wall of a prokaryote, either murin or peptidoglycan. Those are the 2 terms that are used. I always use murin. It's shorter, somehow a bit easier to remember. So now, interesting here, AQA gave you the other option of glycoprotein. I personally would never use that. I always do, look, here we go, accept peptidoglycan and accept phonetic spelling. Not worried about the spelling here. So, name the process by which prokaryotic cells divide. Is it important to know the difference in the structure of flagella in prokaryotes and eukaryotes? For OCRA, cheddar cheese, Dr. Cheddar Cheese, yes, you do need to know it, but I'm going to be really honest, it is hardly ever questioned about that specific detail. For OCRA, you absolutely need to know that the flagella is kind of, it's an arrangement, 9+2 arrangement of microtubules, but that's about it, all right? And cilia also have a similar arrangement. It's kind of a microtubule kind of structure, yeah. So, but if you've got other things to learn, I wouldn't emphasize that bit to learn. I wouldn't prioritize that bit to learn. Hope that makes sense. Name the process by which prokaryotic cells divide. I hope there is a shout out there for binary fission, not mitosis. There are no linear chromosomes, therefore that chromosome dance of mitosis can't happen. It is binary fission. I love the fact that they say don't allow fusion. Fusion means 2 things coming together, not being, you know, separated. So, okay. Some prokaryotic cells can divide every 30 minutes. Oh, here we go. So I got that mark. So another bit of maths. A liquid culture contained 13,500 cells. Okay, fair enough. Assuming each cell divides every 30 minutes, calculate how many cells there will be after 3 hours. So here's the logic. At each division, the number of cells is going to double. If it was 1 cell, it would go from 1 to 2, doubling. Go from 2 to 4, it would go from 4 to 8, 8 to 16, 16 to 32, bloody, bloody, blah. Okay, we don't start with 1. We start with 1.35 times 10 to the 4. Therefore, does it make sense that by however many number of divisions there are, which in 3 hours there will be 6 divisions, there are 6 doublings. 6 lots of times 2, which is 2 to the power of 6. And we haven't started with 1 cell, we've started with 1.34 times 10 to the 4 cells. And then we times that by 2 6 times. And that gives you this rather large number of 8.64 times 10 to the 5, which of course is the right answer. And it could be 864,000 if you wanted to write it like that. You can write it pretty much however you like. Always look out in questions. If they tell you to write it in standard form, make sure you do as you're told. Otherwise, you should be able to get away with it writing it as a whole number. Hope that makes sense. Okay, figure 4 represents a triglyceride. Name the molecules. Well, P is glycerol and Q is fatty acid. There are 3 fatty acids attached to a glycerol, and the bond between each fatty acid and the glycerol is an ester bond. Oh, forgot that. Okay, got those marks. Okay. Describe how you would test a liquid sample for the presence of lipid and how you would recognize a positive result. So, first of all, you would be adding ethanol because ethanol dissolves the lipid. All right, if you add ethanol, whatever lipids there are in the sample will be dissolved into the ethanol. But then you add a few drops of water because that water causes the lipids to come out of solution and to become an emulsion. Sorry, that isn't quite true. The addition of the water causes an emulsion to form between the lipid that is dissolved in the ethanol and the water, and you end up with an emulsion. So there we go. White emulsion is a positive result. So there's the mark scheme. Sequence is important. You cannot add the water first. It doesn't work. Don't like the word cloudy, and notice it is not a precipitate. It's not making something solid out of because of a reaction, a solid product of a reaction out of 2 aqueous solutions. That is not what is happening. So it's not a precipitate. You can't use that word. It's incorrect. Okay, so here is my tiny little plug on Biology Carol. I have a website. On that website, so YouTube pays me a tiny, tiny amount of money. I could not carry on spending the amount of time I do to make videos for YouTube if I didn't have a little bit more income. Just wouldn't be possible. So I have put all of my teaching videos that cover the whole syllabus for AQA and OCRA onto a website. And to subscribe to both year 1 and year 2 of the A-level course for both years, to subscribe to those two years for a whole calendar year, it's £100 for the whole year. That's kind of 2 hours of personal tuition, which I'm hoping is a bargain. If you just need AS, if you're about to start AS, it's 50 quid, 1 hour's tuition for hundreds of hours of teaching. I am absolutely committed to making this accessible for students who cannot afford tuition. That is my goal. And therefore, I've put this in place that you can actually earn your subscription back if you get friends to buy the videos from the website. And there is also a freebie. So if nothing else, just go and have a look at the website and see what you think of it. And if you want to put a little comment on my YouTube, I answer all comments on my YouTube. And on the website, it is basically like Biology Carol YouTube. You can write little comments under the videos, or ask questions about the videos, and I come back to you generally within 24 hours. Okay, onwards and upwards. Figure 5 represents phylogenetic classification. So classification, putting things into groups, phylogenetic, according to how closely related they are, according to how recent their most recent common ancestor is. So phylogenetic classification of 4 different species of fruit fly. Figure 5 shows a hierarchy. Classification, we do hierarchical classification. And the definition of the hierarchical classification is that it is groups within groups and none of those groups overlap. A particular species cannot be part of more than one group. It can't belong to two kingdoms. It can't belong to two phyla. It can't belong to two orders or two classes or two families or two genuses or two species. Yeah. So because it shows smaller groups within larger groups with no overlap between any groups. That is the definition of hierarchical classification. So I get, yeah, groups within groups, absolutely fine. So name, now, sorry, I've got something covering this. Name the taxa that the flies belong to, that Drosophilidae, oh dear, I can't pronounce that, belongs to. I think that's what the question is. Basically, you're working out which taxa we are looking at here. Well, clearly these, and you can tell by the way it is written, the way the binomial name is written, the genus is capitalized, the species have got a lowercase. So therefore, this is the taxa above genus. So if I write, so here's how I remember this: keep pans clean or family gets sick. That's how I remember it. The one above genus is family. So the answer is family. So, okay. Drosophila erecta and Drosophila willistonii are closely related species, but different species. Describe evidence from figure 6 that supports this statement. This is all about courtship behaviour. OCRA students, you do not cover courtship behaviour, so don't worry about this. So here we are told that one of the stages of courtship is singing by the males. Normally the male would produce a sign song in which continual noise is made, and then there is a pulse song in which there is continual noise with some louder peaks. Scientists showed fruit flies a visual stimulus that made them sing. They made recordings of the songs. Okay, lovely. Here are the recordings. Lovely. So, describe evidence that these two, are closely related but different. So this is what I wrote. So first of all, number one, I was going to compare the two male songs. Clearly, it doesn't make a lot of sense to compare the female song of one species with the male song of another. Let's only change one thing. So here am I comparing the males, and I noticed that the sign songs are similar lengths, as are the pulse songs. And that implies that they are closely related. However, the pulse songs differ. And the most obvious thing to me that I noticed that actually the interval between the louder peaks were different between the two species, which is why it suggested different species. And apologies that I've spelt that wrong. But hopefully that answer makes sense. But notice that I really look carefully at the pattern of this courtship song to really pick out the things that are similar, or to describe the things that are similar and the things that are different. Domain, yes. Yes, I can see. So domains are kind of above kingdoms, but given that the previous question wasn't talking about domains at all, we were clearly down at the dare I say it, the smaller taxa. Yeah, we weren't talking up at the kingdom, phylum, domain level. We were, all of those ones that were being talked about in that previous question were down at the obviously species, genus, family level, which is why I didn't put domain in that little what do you call it? Acronym? No, mnemonic. Explaining that. All right, so I can talk about domains and kingdoms in a minute. Okay, all right. So I got the 2 marks as a result of that. The scientists repeated their experiments using female fruit flies as the visual stimulus. When a male and a female Drosophila willistonii were together, their songs led to mating. When two female Drosophila willistonii were together, their songs did not lead to any attempt to mate. Use information from figure 6 to explain why the females did not attempt to mate. So I kind of went, okay, we're looking at the difference between the female and the male here. Well, clearly the female would have recognized that the male song, oh sorry, the male will have, the females will have recognized that the male song is different to another female. So I said, because the female pulse song has a longer interval between the first and second louder peaks compared to the male, therefore the females knew that there was no male present. That's what I wrote. So here is the mark scheme, and you can see that I didn't get both of the marks. I only got the first mark, kind of pointing out they were different, because I didn't quite finish nailing the end of the question to kind of finish off saying, therefore it did not mate. There wasn't a stimulus to cause mating. I didn't write it. I missed the mark. Not going to get too worried about that. So question 7. Question 7. What is the proteome of a cell? The proteome of a cell are all the proteins that can be made by that cell. And this is what I wrote. This is what the mark scheme said. Obviously, my answer here is talking about all of the proteins rather than talking about all of the proteins that the DNA is able to code for. I didn't talk about DNA, but I got the mark. Give 2 structural differences between a molecule of messenger RNA and a molecule of transfer RNA. So the first thing in my mind is that messenger RNA is a linear single-stranded molecule and transfer RNA is a cloverleaf-shaped single-stranded molecule. Oh, and obviously that was the one that I put second. Oh yes, and there the other thing, the most obvious thing that I talked about was that tRNA, that cloverleaf shape, has both an anticodon at the bottom of its structure and an amino acid binding site at the top. And mRNA has neither. Here's the mark scheme. So here is mark point 1 here. I talked about this one here. And I also nailed that the mRNA doesn't have an amino acid binding site, but tRNA does. Notice that the whole anticodon-codon comparison actually was in mark point 5, and I clearly didn't say that mRNA has codons instead of, I talked about them either being present or absent. So, but fortunately, I think I would have got those 2 marks. Now, the interesting thing here is that they ask for 2 structural differences. If you, I wasn't trying to put in more than 2. I simply chose a different way of comparing them. So I believe I would have got the 2 marks. If you are obviously not writing 2 differences, but you are writing 3 or 4 differences, basically trying to hedge your bets, the rule of thumb is for examiners is that they will mark the first 2 and then they will ignore the rest. No, in fact, they won't they will mark the first 2. So it's not worth you trying to write more than, that is why you can sometimes be penalized if you give more than 2 answers. Because also, if you give more than 2 answers, and let's say answer 3 or 4 was incorrect, you would have a mark deducted. The wrong answer would counteract one of your correct answers. So if they give the instruction only to, do as you're told. All right. I hope that makes sense. Okay. Starting with mRNA in the cytoplasm, describe how translation leads to the production of a polypeptide. This is a flipping pig of a question, because translation, I think, is one of the hardest processes to describe clearly. I know in my video on this, on my live stream on this, which should be on YouTube, I think it's still public, I actually give a kind of mark scheme description of translation. The question's very clear. Do not include transcription and don't include splicing. They're not interested in that. Okay, so nicely clear about what to include. So here is my answer. And I basically followed my guidelines that I put on my previous live stream as to what to write in a translation answer. So the ribosome attaches to the first two codons of the mRNA. tRNAs, and I'm going to be honest, this sentence is not the clearest, but I think it's just about okay. tRNAs carrying specific amino acids bind by complementary base pairing. Notice that I'm getting that information in there. Of their anticodons, I'm clearly talking about the tRNAs, of their anticodons to the first two codons on the mRNA. I think that's actually pretty clear, even though it's a little bit of a cumbersome sentence. The ribosome, using the hydrolysis of ATP, let me tell you, I got to writing ATP and I just thought, oh my goodness, just for a few extra characters on the page, I can describe the hydrolysis of ATP. And that is generally a good thing to do. If you know that with just one or two extra words, you can add a little bit more detail, then do it without kind of going overboard. This is why when I was writing the word using, because I was just going to put using ATP, I decided to write using the hydrolysis of ATP, and then I ended up describing the whole thing. Can I just say, if I'd edited this, if I'd really properly edited this, I could have, I probably just should have written using ATP arrow ADP plus PI, because that says it's being broken down. It is the chemical reaction that describes the hydrolysis. Okay, so I'm just, pointing out that you can see here that I have actually written what I would have written in the exam and not finally edited this answer for this video, or for this live stream. So ribosome binds the first two amino acids with a peptide bond. I knew that that was going to be important to get in there. The ribosome then moves along one codon where the next tRNA has bound. I've already described it, I'm not doing it again. Ribosome binds second and third amino acid. I'm now running out of space. Notice my writing is really large in proportion to the space here, though. You'd have much more space. This is repeated until stop codon on mRNA is reached, and then the polypeptide detaches. Here is the mark scheme. And here are the mark points that I got. Mark point 1, because it associates with the ribosome. Mark point 3, because tRNA brings a specific amino acid. Anticodon codon, mark point 4. Mark point 6, probably the whole peptide, amino acids joined by peptide bonds. And here is mark point 5. Ribosome moves along to next codon. Now notice I said ribosome fits around two codons. I could have possibly given myself mark point 5 here. I could have given it either here or down here. I've given it down here because of the way that I worded this answer, because I said the ribosome then moves along one codon. Notice that that doesn't mean the next codon. I could have implied that actually the ribosome was moving backwards. But notice my next few words were the next tRNA. That then is enough to really say to the examiner, or it would have been enough to say to me as an examiner, right, they mean moved on to the next codon. And that's why I gave myself the mark point after this, the tRNA word. I just want to explain that because I think it's important for you to understand that what you write has to absolutely nail what is in the mark scheme, even if you don't use the same wording. Okay, and so I'm just trying to explain that. State and explain the property of water that can help to buffer changes in temperature. Well, that is because it has a high specific heat capacity. And because I know the proper definition of specific heat capacity, I use it to explain it. This means that it takes a lot of energy, because I don't have to know the number, don't have to know that, it takes a lot of energy to heat a kilogram of water by 1 degree C. This is because of the numerous hydrogen bonds between, because I was thinking, explain, how much explanation do they actually want? Hadn't looked at the mark scheme, I didn't know what was in the mark scheme. So I was absolutely confident that this was mark point 1, and it was. So didn't have to know numbers. Can gain, lose a lot of heat, or takes a lot of energy to change the temperature. Yeah, I think that wording is, yeah, it requires a lot of energy to increase the temperature, and actually a lot of energy is taken away from the water when the water temperature decreases. Anyway, let's not be pedantic. Oh, right. Name the 2 products of ATP hydrolysis. I noticed here that the instruction was to name. I made the decision not to use abbreviations because I have seen on a previous AQA paper that where they had asked for the name, they did not allow, they did not give the mark if you used abbreviations. So I gave the names: adenosine diphosphate and inorganic phosphate, which were the right answers to get 1 mark. Notice, however, what was allowed on this paper, and I'm really sorry that this isn't complete consistency between papers. Personally, I would always, whenever I see the word name, I mentally now go, I'm not using abbreviations, I'm doing the whole name. Notice that if you had written adenine diphosphate, you would not have got the mark because it is wrong. Adenosine is the adenine and the ribose sugar together. That is adenosine, which is why adenosine, it is called adenosine diphosphate. So, yeah, okay. Hydrolysis of ATP is catalyzed by the enzyme ATP hydrolase. Yes, a student investigated the effect of ATP concentration, not ATP hydrolase, ATP, so the substrate concentration on the activity of ATP hydrolase. She used shortening of strips of muscle tissue caused by contraction as evidence that ATP was being hydrolyzed. Yeah, because ATP is required for muscle contraction. Took 4 slides, A, B, C, and D, added strips of muscle at the same length to each slide, then added the same volume of different concentrations of ATP solutions to the 4 slides and left each slide for 5 minutes, and then recorded the length of each strip of muscle tissue. Now, this is about the effect of substrate concentration on enzyme action. So other than those given, name 2 variables the student should have controlled. Here am I thinking, what else affects enzyme action? And the 2 obvious ones to me are temperature of the muscle tissue. So here am I envisaging the situation. So the muscle tissue is on the slide, the temperature of the muscle tissue and the temperature of the ATP solution being put on to the muscle tissue. And also the pH of the ATP solution. All right, you're not going to control the pH of cells because otherwise you're going to screw up the enzymes in those cells, yeah? but it does matter the pH of the ATP concentration. Okay, so, oh, sorry, I've just realised that I've missed some. Let me just say I've got those 2 marks. Notice that also you could have absolutely talked about the fact that the muscle strips needed to be either from the same species or from the same type of muscle. And I've just realised that okay, I've got no idea what's going on in the little messaging. There's a little chat going on, which I'm not completely understanding, and I'm going to leave you to it. Okay, let us move on. Describe and explain the patterns shown by the data. Now, here's the thing. This is an AS question, so they are being really kind to you and giving you separate bits of the answer sheet for the description and for the explanation. If this was an A-level standard question, without doubt, they would just write describe and explain the pattern, and you would have to work out the difference between the two. Describe is saying what something looks like. You know, kind of tell the story, say what is happening. Explanation is why or how something is happening. So the description is that as ATP solution concentration increases, the final length of the muscle tissue decreases. The explanation is, well, my thought was that there was more ATP available as the ATP concentration increased, and this meant that more muscle contraction could occur and therefore more shortening of the muscle. Here is the mark scheme. Got mark point 1, but I didn't get mark point 2 because I did not include this bit of the mark point. So more energy is released. I thought I'd written enough, but I clearly hadn't. Notice, as always, you can never write about energy being produced. Energy can neither be produced nor, it can neither be created nor destroyed. So it can't be produced, it can't be made. Okay, another bit of calculation. The hydrolysis of 1 decimeter cubed, a decimeter cubed equals 1 litre, and that equals 1,000 centimeters cubed. Always useful. I always picture a decimeter cubed as being a litre carton of orange juice, for example. I always have that in my mind. And I know that that's 1,000 centimeters cubed. So hydrolysis of 1 litre of a 1 molar solution of ATP, where there is 1 mole per decimeter cubed of ATP, releases 30,500 joules of energy. Lovely. 60% of the energy is released as heat. The rest is muscle contraction. So only 40% of this goes to muscle contraction. The student added 0.05 centimeters cubed, not 1,000 centimeters cubed, 0.05 centimeters cubed of ATP solution to slide D. Slide D has a concentration of 8 times 10 to the negative 6 moles per decimeter cubed. Calculate the energy available from ATP for muscle contraction on that slide. So we have got to work out a whole load of fractions. We've got to work out the fraction of 0.05 out of 1,000, 0.05 out of 1,000. We have also got to work out the fraction of 8 times 10 to the minus 6 out of 1. Here am I, I'm writing this, trying to make it clear, and timesing that by the number of joules that would be released. But then we need to times it by 0.4 or 40%, 40 over 100, because that is the percentage, that is the proportion that actually goes to muscle contraction. But just notice you have to work out certain fractions, because only a tiny fraction of the volume and a tiny fraction of the concentration has been used. and only a fraction of the energy is available for the muscle contraction. And that gives you this number on your calculator. Now, personally, I would just convert this to standard form. So because the decimal place is moving 1, 2, 3, 4, 5, 6 places, this is going to be 4.88 times 10 to the minus 6, because we're talking about a fraction of 1. Okay, there we go. That's how many. And that is the answer. And I got all 3 marks. Notice they allowed you to round it up. Except correct answer, however expressed, they would have allowed me to give this answer. Okay, hello. Describe the pathway taken by an oxygen molecule. We're nearly there. Oxygen molecule from the alveolus to the blood. Well, I absolutely know this. Notice that we're starting in the alveolus, from the alveolus to the blood. There are only 2 layers of cells that the oxygen molecule goes through. It goes through the alveolar epithelium and the capillary endothelium. Remember that single layer of cells that makes the capillary wall is called the capillary endothelium. So here is, I'd never call it the epithelium of a capillary. I would always call it the endothelium. So anyway, I got the 2 marks. Oh, you've got to get the sequence. You've got to get, because you've got to take in that you're going from the alveolus to the blood. So there we go. Explain how one feature of an alveolus, not of the lung, i.e. millions of alveoli, explain how one feature of an alveolus allows efficient gas exchange to occur. Well, to be honest, there is only one thing that you can talk about of a single alveolus that allows efficient gas exchange, and that is the fact that the epithelium is 1 cell thick. Efficient gas exchange, I always think Fick's law. That is that the rate of diffusion is proportional to surface area. That doesn't apply because we're only talking about 1 alveolus, times the concentration gradient. That doesn't apply because we're talking only about an alveolus, not the alveolus and the blood flow going past, which of course is what maintains the concentration gradient, and divided by the length of the diffusion pathway. And this is what's being talked about here. The alveolar epithelium is 1 cell thick, and this provides a short diffusion pathway, which increases the rate of diffusion. The shorter the diffusion pathway, the higher the rate of diffusion. So, notice here, this is such a common error. People talk about a thin membrane. Well, a membrane is what is on the outside of a cell, and a membrane is the same flipping thickness everywhere. It's a phospholipid bilayer, fundamentally. You're not going to get membranes of different thicknesses, so be careful about your terminology. I'm going, Jason, planning to go over paper 2 next week. There's the plan. So yeah, paper 1 this week, paper 2 next week. Okay, in England, in October 2015, a law was introduced making it illegal to smoke in a car carrying someone who's under the age of 18. Following the introduction of the law, politicians have stated, it is dangerous to smoke in a car when a child is in the car. Higher levels of deadly toxins can build up even on short journeys, and children breathe faster than adults, meaning they inhale more of the deadly toxins. Use the information provided and the data. So use the information provided, that is this information here, which we're about to go through, and the data provided, which of course is the graph, to evaluate the politician's statements. Evaluate means for and against. Oops, sorry, yes, so let me go through the data. Let me go through the information. The carbon monoxide is in cigarette smoke. It binds with hemoglobin. That's its issue. It binds instead of the oxygen, and therefore the blood is carrying carbon monoxide instead of oxygen. The body is depleted of oxygen, cells die, potentially the person dies if they're in carbon monoxide zones, environments for too long with high enough concentration. Scientists investigated the concentration of carbon monoxide in cars in which people were smoking or not smoking. Here's your non-smoking, here is your smoking lot. They measured the concentration with the car doors, windows open, white bars, and closed, black bars. Figure 7 shows the scientists' results. 2 standard deviations either side of the mean, which includes over 95% of the data, which you probably should know. Okay, so looking at this data, I'm already noticing that there is no significant difference between any of these groups if the window is open, which kind of makes sense because there's absolutely ventilation. The carbon monoxide is swept out of the window. There's no significant difference because the error bars are overlapping. Now, there is a significant difference between a car, both situations where the windows are closed, not terribly surprisingly, in comparison to in a car where no smoking is occurring. If there's been smoking for 5 minutes in the car, there is a significant increase. With the windows closed, there is a significant increase in the carbon monoxide concentration in the car, as you can see, because these error bars do not overlap. And also 5 minutes after the smoking, funnily enough, if the windows are closed, the flipping carbon monoxide is still in the car. So there is still a significant difference to no smoking. So this is what I wrote. All right, here we go. Use the information provided. And here am I reading the question carefully to make sure I'm doing what I'm flipping told. I'm not just going to talk about the data. I'm also going to pull in some of the information. To evaluate means a plus or minus pros and cons, for and against. So this is what I wrote. I could have written pros or for and against. It's actually quite a useful way to separate out your answer because evaluation is always for and against. So arguments that how the data supports these statements. Well, there's a significantly different mean concentration of carbon monoxide in the car when the windows are closed. So here am I spelling it out, writing what is in the data. After 5 minutes of smoking and 5 minutes after smoking when compared to non-smoking. Shown by, how do I know this? Shown by no overlap of the standard deviations of these groups. Then I'm kind of going, right, how can I talk about this? Oh, the toxic carbon dioxide builds up with closed windows. Here am I referring to, toxic, the toxins build up with closed windows and would bind to hemoglobin in place of oxygen. Couldn't really think of anything more to add. Against. There is no significant difference in the 2 smoking groups. I'm now realizing that I'm running out of space, so I'm trying to cut down what I'm saying. Notice that my writing is relatively much larger, I think, than yours would be on an exam paper. There is no significant difference in the 2 smoking groups compared to no smoking when the windows are open, as the standard deviations overlap. Children, and here I want to kind of talk about this because this also, children breathe faster than adults. Well, yeah, okay, even if they do, and I'm not even sure, they probably do, don't really know. Children breathe faster than adults. They've got much smaller lungs, yeah? So children may breathe faster, but their lung volume is smaller, and therefore the amount of carbon monoxide inhaled would potentially be similar. And as I read that, I kind of thought, that doesn't make a lot of sense to me, so I pointed that out. So here are this is the mark scheme. Are there any? Yes, and here are the notes. So here are where I got the marks. I got a mark here talking about, they didn't require me to go on and on about the 2 smoking groups, you know, anyway. I got mark point 2 here. It's because carbon dioxide causes less oxygen to be carried. I've said it binds in place of oxygen. By definition, that means that less oxygen will be carried, surely. And then we have got mark point 6. Oh, no, not mark point 6. Which one is that? When windows are open. Oh, mark point 5 down here. And finally, mark point 6 here. Oh, come on. Isn't that working? Mark point 6, here we go. Okay, so I did end up with the 4 marks. Good point here that actually I hadn't made any reference to the fact, who flipping knows how high the concentration has to be for it to be dangerous? You know, I thought that was absolutely a valid point. And also, obviously, you know, there's no data on child breathing rates. So yeah, there was a number of things you could have talked about. I think this is the last question. Okay, I'm actually, I don't want to summarize this passage. Okay, let's summarize it. Right, sizes of population of normal intestinal bacteria. Our intestines are full of normal bacteria, healthy bacteria, are normally controlled by T cells that are produced slowly and in small numbers by the immune system. They don't survive for very long. As a result, they don't release large amounts of cytokines. So the amount of cytokines is down to basically the number that there are in the system. Cytokines are chemicals that can cause swelling in the lining of the intestines. Crohn's disease is a long-lasting disease caused by the swelling of the lining of the intestines, all right? And it's caused by a loss of tolerance to normal intestinal bacteria, as shown by an unusually large response by T cells. This response can be triggered by pathogenic bacteria in the intestines of people with a genetic tendency to get Crohn's disease. So what they're saying is, somebody with a genetic tendency to get Crohn's disease, if they get a pathogenic bacteria in their intestines, that can kick off massive amounts of T cells. And then those T cells actually become intolerant to the normal gut bacteria. And therefore, they carry on being produced as a result of the normal gut bacteria, even though the pathogenic one has kind of disappeared once it's been got rid of. I hope that kind of makes sense. Some people, Crohn's disease can be controlled by a drug called 5-ASA, which reduces swelling, presumably by reducing the number of cytokines, by reducing the number of T cells. Another drug called 6-MP can also be used. It inhibits an enzyme required to make adenine and guanine. These are the bases that are used in DNA replication. This is effective because most cells can recycle nucleotides, but T cells are not able to. Use information from the passage and your own knowledge to answer the questions. Crohn's disease symptom of swelling of the lining of the intestines could be triggered by pathogenic bacteria in the intestines. Suggest how. This is your immune response stuff. Oh, sorry, I forgot that was my highlighting. So this is what I wrote. Pathogenic bacteria will have specific antigens on their surface, their cell surface. These will bind to complementary receptors on T helper cells, causing clonal expansion by mitosis. Because the pathogenic bacteria cause an unusually large response by T cells, many more T helper cells are produced and many more cytokines are also produced. And these are what cause the swelling, equals Crohn's. So here is the mark scheme. Here are the examiner's comments, and this is where I got the marks. Talked about the antigen. Notice that it's always the antigen that kicks off T cells. If you're asked about T cells or B cells, nearly always bring that word antigen into your answer. It took me a long time to get my second mark. Causes more T cells to be produced, and therefore more cytokines, which cause more swelling. So I didn't get, sorry, I think, yes, this should have actually been mark 0.5, not mark 0.3. Didn't get mark 0.3 or mark 0.4, but I got the 3 marks. Suggest the meaning of a genetic tendency to Crohn's disease. Well, that to me means that somebody has particular alleles in their genetic makeup. Notice not particular genes, because every human shares the same genes. So individuals carry alleles of certain genes that leads to an unusually large response of T cells to pathogenic bacteria. Here is the mark scheme. Some people have a mutation. Well, mutations cause alleles. I take exception to this. I don't understand. Have a gene. We all have the same genes. We have different alleles. That's why I have not used the word gene. I've used the word alleles. I actually would not sure that is correct, but anyway, and I didn't, to be honest, I didn't know what else to write after this. I didn't think I was going to get the second mark. That increases the chances, risk. I didn't talk about chances, didn't talk about risk. Makes it more likely, didn't say that. Causes them to have an unusually large T cell, leads to the unusual. I think I got it by the skin of my teeth. I think I got both the marks. Suggest why 5-ASA is only effective in controlling the swelling of the lining of the intestines in some people. I had to read this several times. Some people, Crohn's disease can be controlled by a drug called 5-ASA that reduces the swelling. Well, that to me means that it has to be able to counteract the cytokines, because cytokines cause the swelling. So I did wonder, given that it talked about an unusually large response, I just thought, well, maybe some people produce so many cytokines that there isn't enough 5-ASA just can't. But I was not convinced by this answer at all. And I was actually very surprised that I ended up getting those marks. I was not convinced that that was going to be right. I certainly would never have thought of this whole allergic response. I wouldn't have thought of that. And finally, suggest why 6-MP can be used to control the symptoms of Crohn's disease. Do not include details of enzyme inhibition or protein synthesis in your answer. Do as you're told, because otherwise you will be wasting your time and not having any hope of getting any marks. So 6-MP inhibits an enzyme required to make adenine and guanine. This is effective because most cells can recycle nucleotides, but T cells can't. This is what I wrote. If 6-MP reduces the production of adenine and guanine and T cells are unable to recycle them, then the T cells will have less adenine and guanine. Therefore, notice I'm now following my argument on. Therefore, less or no DNA replication can occur. And therefore, less mitosis can occur. Therefore, less T cells are produced, therefore less cytokines, therefore less swelling. I mean, I literally, talk about making my point. Here is the mark scheme. And here is, and here are the examiner's comments, and here are where I got the marks. Notice it took me a long time to get the first mark. Lack of adenine and guanine will slow DNA synthesis. Less T cells are produced, and therefore less cytokines are produced. And therefore I got the 3 marks. Okay, and there we are finished. So unless, I'm hoping that that has been useful. Next week we will go over AQA paper 2. If you have any questions, start typing now rather quickly. If you have a question, just put yes in, because then I will hold on until you have actually typed your question. But otherwise, I am going to say so long and farewell until next Tuesday, when we are going to go over AQA paper AS level paper 2 from 2018. And I look forward to seeing you then. I think so far we haven't got any questions. So I'm going to say, oh, thank you, Simone. I'm going to say goodbye and hopefully see you next week. Thank you for the thank yous, guys, and see you soon. Bye-bye.